The title of the paper that I will be presenting is Exercise Training Improves Vascular Mitochondrial Function, written by Song Young Park et al., published January 29th of this past year. So we're going to quickly go over some background to allow us to further understand the results of the paper. So the amount of mitochondria in a certain cell depends on the tissue that it is a part of. So for example, the heart has high metabolic rate and therefore will have more mitochondria than the vasculature. So therefore, the amount of mitochondria in a certain cell is tissue specific. When normalized, the mitochondrial respiratory capacity in the three different types of muscles are similar. Furthermore, exercise training has been shown to improve cardiac and skeletal muscle mitochondrial respiratory capacity, where the respiratory capacity is the ability for the mitochondria to undergo cellular respiration. It is thought that a small density of mitochondria within the endothelium may serve as signaling organelles that receive signals from the local environment. Nitric oxide, or NO, is generated by enos and has been known to serve as a signal to cause for an increase in mitochondrial respiration in different cells. If NO's production is limited, there are physiological interventions that can possibly increase enos and therefore NO generation. And some of these physiological interventions may be as simple as exercise training that can then facilitate vascular mitochondrial respiration. So the main hypothesis for this paper is that exercise training concurrently increases both vascular mitochondrial respiratory capacity and vascular function, with vascular being defined as anything relating, affecting, or consisting of vessels that carry blood. So increasing vascular function will therefore increase the amount of oxygen and nutrients that can get circulated throughout the blood and to the rest of the body. So the purpose of this experiment was to determine the impact of exercise training on vascular mitochondrial respiratory function. Possible insight into potential mechanisms and consequences of exercise training induced mitochondrial and vascular adaptations was also of focus. So for the experimental design, these researchers took 6-week-old male mice, took those mice and split them into two groups, with one of the groups being sedentary or SED mice and the other group being swim-trained or EX mice. The EX mice completed 2 swimming training sessions per day, with each session being separated by at least 4-hour intervals for 36 days. The SED mice also swam twice a day, but stayed in their cages. The mice were then killed, and their aorta, iliac arteries, and femoral arteries were dissected and removed for research. So they had a variety of means and methods to get the data that they wanted to analyze and see if it was consistent with their hypothesis. For one, they measured the vascular mitochondrial respiration by looking at the aorta and measuring the oxygen consumption rate. To check to see if the integrity of the outer mitochondrial membrane was intact, the researchers injected cytochrome C to see if complex 1 and 2 of the mitochondria were working properly, as cytochrome C is associated with those two complexes. Second, they looked at the measurement of mitochondrial DNA content. DNA was taken from the dissected aortic segments and dissolved so that it could be subjected to real-time PCR amplification. Measurement of vascular function was the third thing they looked at. To test for vascular function, the femoral arteries were looked at. Isometric tension techniques were used to determine the vessel diameter that displayed the greatest tension development. For this figure, parts A, B, and C, the solid black column illustrates the SED mice and the white columns illustrate the EX mice. Part A illustrates that citrate synthase activity in both the SED and EX mice. Citrate synthase is an important component of cellular respiration, and it is the first enzyme that is a part of the citric acid cycle of cellular respiration. Since it is a part of cellular respiration, we would expect its activity to be high in cells that are able to undergo cellular respiration. This is seen in Part A, as both SED and EX mice show citrate synthase activity. Furthermore, it is seen that EX mice show higher levels of activity of citrate synthase than the SAD mice. This significant difference shows that the mice who were subjected to exercise were able to have higher amounts of cellular respiration. Part B illustrates mitochondrial oxygen consumption in aortas from EX and SED mice. There are 3 different groups of mice represented here. Complex 1, State 2, Complex 1, State 3, and Complex 1 and 2, State 3. The complexes refer to the protein complexes that are along the inner mitochondrial membrane that are responsible for oxidative phosphorylation. State 2 represents the time when ADP is added and respiration increases only modestly, which means that State 2 has a slow respiration rate. State 3 represents the time that the respiratory substrate, for example, pyruvate, is added, which causes respiration to increase markedly and allows for a fast respiration rate. As the process goes from complex to complex and from state to state, the amount of cellular respiration that occurs increases as well. This is illustrated in both the SED and EX mice, as the respiration rate increases as we move from left to right. Additionally, the EX mice are seen to have a higher respiration rate than the SED mice, with the later 2 columns having a significant difference between the 2 groups. Part C illustrates the respiratory control ratio, or RCR, that is a useful general term of measure to see the function in isolated mitochondria. High RCR means that the mitochondria is functioning pretty well, while low RCR means that the mitochondria is not functioning well. In this case, the EX mice has a significantly higher RCR value than the SED mice, which illustrates that exercise helped in the functioning of the mitochondria. Secondly, we're going to look at how vascular indices of antioxidant capacity follows exercise training. For Part A, we see a Western blot that illustrates MNSOD protein expression in comparison to a normalized GAPDH expression. MNSOD is a protein that stands for mitochondrial antioxidant manganese superoxide dismutase, which means that this protein is activated in the mitochondria to detoxify and get rid of free oxygen radicals that are a byproduct of cellular respiration. When comparing the image for SED and EX mice, we see that the MNSOD band for the EX mice strain was darker and more prominent than the SED mice. Since there are more MNSOD present in the EX mice, it illustrates that there is more MNSOD activated to then eliminate the free oxygen radicals that are being generated from the increase in cellular respiration. This is further illustrated in Part B, where the EX mice are significantly higher in terms of the arbitrary units that measure for MNSOD and GAPDH. Part C illustrates the reactive oxygen species that exists. DCFDA stands for dichlorofluorescin diacetate, which is a fluorescent protein to measure the amount of ROS. The significant decrease in ROS in the EX illustrates that the exercise-trained mice are better at eliminating the amount of ROS species present within its cells of the artery that it was dissected in. In conclusion, these results indicated that exercise training increases vascular mitochondrial capacity and it also improves vascular redox balance. This is important since it shows that there is a potential to protect against age-related and disease-related challenges to arterial function. So the overall message that I got from this paper was that exercise training and physical activity is important to maintaining and or improving vascular health. So, in other words, when people tell us that we need to exercise, there's a reason to why we need exercise, because exercising allows us to maintain our vascular health and allows for our circulation to be up to date and even better. So the 2 questions that I have are, in addition to looking at the effects of the mitochondria and arteries, what are other ways one can see the effects of exercise on the body? Secondly, what would the results look like if the experimenters used differently aged mice within the SED and EX subgroups?